So it's a great pleasure to introduce Goujun Bu, who is going to be our speaker this morning. Dr. Bu obtained a bachelor's in biology in Beijing, followed by a PhD training in biochemistry from Virginia Tech and experience in molecular biology and neuroscience. He established a terrific research career in cell biology and neuroscience at Washington University, where he was awarded the rank of professor. And then he moved to Mayo Clinic in 2010, where he has continued to advance research in a number of areas, in particular in relation to APOE biology, and also looking at uses of fibroblast-derived stem cells in biology and potentially with therapeutic implications. Dr. Bu has published very widely in first-rate journals and has received many honors and awards. These include the Faculty Scholar Award and the Zenith Fellows Award from the Alzheimer Association, and also an Established Investigator Award from the American Heart Association. I can say a lot more about Goujun, but in the interest of letting him do the talking and tell us about the exciting biology and some new findings on APOE, I'm going to throw things open to Goujun. So welcome.
Thank you, Doug. Very generous introduction. As Doug was saying, that I've been working on APOE for many years, actually starting 1993 when APOE was discovered as a risk gene for Alzheimer's disease. So that was the latter part of my postdoctoral training, working on actually APOE receptor LRP1. And more recent years, the lab has been focusing on more on APOE itself. So what I'd like to do today is really just try to give you first a broader view where we are in terms of understanding how APOE impact Alzheimer's risk, with different pathways involved, then focus on mostly unpublished data on, how we're doing the molecular profiling, looking at the APOE effect in different tissues or cell type, using, as Dr. mentioned, mouse model and also iPSC model. So I think this audience, I don't have to say too much about Alzheimer's disease, but simply say it's defined by the extracellular A beta plaque and intraneuronal tau tangle. These are pathological lesions collectively together define Alzheimer's disease. It actually does not necessarily require the presence of neurodegeneration so that the disease could be preclinical. Obviously, many years ago, we had to rely on autopsy brain to see these pathological lesions, but now we have imaging and fluid biomarker tools to really detect, the presence or extent of these pathology. Just a few quick facts about Alzheimer's disease. It's not normal aging. It's a disease. It does kill people. Aging is the biggest risk factor, and with APOE4 being the strongest genetic risk factor, but obviously there's so many factors. And then we really haven't had a disease-altering modifying drug really until last summer. There's this aducanumab approved, even though there's a lot of controversy in terms of the actual efficacy on the cognitive improvement or slowdown. So in Alzheimer's brain, in addition to plaque and tangle, we also have other mixed pathology, for example, the vascular pathology, cerebral amyloid angiopathy, but also about half of the Alzheimer's brain had TDP-43 pathology, and then about another half have the alpha-synuclein pathology and synaptic loss and neurodegeneration. Obviously, these are the driver for dementia. So really the field of Alzheimer's disease has been really led by genetic discovery. So first in the early 90s, here's a discovery about mutation in this three gene, APP, presenilin 1 and 2. Mutation of these genes cause familial form of early onset disease, dominant inherited. And because they all collectively increase A beta, in particular, the more aggregated form of A beta 42, this allowed the proposal of so-called amyloid hypothesis. Then in the last 20 years or so, there are genetic genome-wide association study where some of these risk genes, they typically have smaller effect size, but quite common in general population, were identified. And now there are about hundreds of them defined as AD risk gene. These are some of the genes that we have interest. But these genes are typically mapped to three very important biological pathways: lipid metabolism, in particular cholesterol, membrane trafficking, and also innate immunity or microglial biology. And then in the last 10 years or so, we moved to the genome sequencing age, where some of the rare variants, not 100% penetrating, but really rare variants were discovered. TREM2 was one of them, discovered late 2012. This is very important for TREM2 discovery because this particular gene expressed only in microglia in the brain. So really bring the microglia to the center stage of Alzheimer research and also collectively innate immunity. Then you have APOE. So APOE is one of these genes that's actually quite common with the risky alleles E4. General population of 13% allele frequency, over 20% of people carry at least one allele of E4, but also have relatively high effect size. So what's interesting, I'm going to tell you some of this today, is that APOE4 impact all these four major pathways: amyloid hypothesis, lipid metabolism, membrane trafficking, and innate immunity. So I think maybe that's one of the reasons why E4 such a strong risk, and really present an opportunity for us both to understand the disease and also, now many people from pharmaceutical company, biotech, they're interested in targeting APOE because it has an impact beyond just amyloid. So just a bit about APOE. I mentioned about E4 is the strongest genetic risk factor. One allele 3 to 4-fold increase of risk, and two allele 10 to 15-fold. APOE2 is protected. In terms of biology, APOE is one of these so-called apolipoprotein, where it carries lipid from cell to cell, tissue to tissue. So in this case, APOE normally adopt an enclosed structure. Lipid binding to the C-terminal domain typically opened up this structure, where the N-terminal domain had this 4-helix bundle, including the receptor binding region, receptors such as LRP1, LDL receptor, heparan sulfate proteoglycan. In this fashion, APOE will deliver the lipid cargo to the target cells. So it's very straightforward. APOE does this in periphery through chylomicron VLDL, subclass of HDL in the brain, primarily from astrocyte to neuron, but also microglial oligodendrocytes as well. So discovery in 93, now almost 30 years, what have we learned about E4 being a risk factor for Alzheimer's disease? How does it drive the disease risk? So I would say we know quite a lot. We don't know everything or really the specific contribution each pathway, but we do know a lot. And I think most of us in the field agree that the major pathway, the APOE4 impact Alzheimer's risk, is driving the early and more abundant amyloid. And it does this by inhibiting A-beta clearance and promoting A-beta aggregation. Okay. But APOE also have effect on the vasculature, for example, enhance blood-brain barrier leakage in an age-dependent manner, impair vascular function, also impact metabolism. So glucose, mitochondrial function, insulin signaling. It also could affect directly of tau tangle or tau-mediated neurodegeneration. But if you look at actually APOE co-aggregate with A-beta in the amyloid plaque, so the functional pool of APOE4 is actually smaller than E3 or E2. And as I mentioned, APOE does have an important biological function, which is lipid and cholesterol metabolism. So that lead to the loss of function of APOE4 in its normal physiology, carrying lipid, also for microglial inflammatory pathway. And some of these are important for injury repair, synaptic remodeling, and so on. So I think these thinking of either gain of toxic function, loss of function, sometimes can guide us a little bit in terms of how do we think about targeting APOE. So amyloid is the main pathway. Just show a little bit of the evidence the field had. This is one of those meta-analysis of amyloid PET data. Meta-analysis, if you look at the individual who have two allele of E4, E4, you can see that these individuals, these actually normal cognitive individual, they have amyloid development already in their 40s, and then they keep moving up, right? So if you look at the MCI population in the same publication, it follow the same profile or similar profile. If individual have one allele E4, it almost does not matter whether the other alleles are E3 or E2, they have very similar amyloid profile. You know, mostly in the 60s, they develop amyloid. And then in this fashion, E4 is actually dominant in driving the amyloid because E2 is not protective in this case. If there's no E4 allele, E3, E3 has less amyloid, then E2 is protective. E2, E3 have less amyloid in 3, and then E2, E2 obviously smaller number, and no detectable amyloid by this PET imaging. So over the years, my lab and others have really tried to study or understand the mechanism, how APOE receptors impact A-beta clearance or aggregation. And this is one of those studies that we published a few years back, where we found that if you knock out a gene called EXT1 that eliminate the heparin sulfate on the cell surface, you accelerate the A-beta clearance and decrease the amyloid deposition. And then because this allows the normal quote unquote clearance pathways such as LRP1, astrocyte microglia, extracellular degradation function more efficiently because both A-beta and APOE actually they bind heparin sulfate. And with another thing in the field that maybe through this fashion, heparin sulfate serve as a reservoir for A-beta aggregate. And in fact, likely tau and alpha-synuclein also aggregate in this fashion. Mark Diamond and others have shown that heparin sulfate proteoglycan have role in tau propagation from cell to cell. So one of the questions we want to address is what is the mechanism that E4 drive the amyloid pathology? And so to do that, we developed this very versatile targeting allele to generate conditional animal model. So the few feature about this allele is that it's a Cre-Lox system. So normally there's a flock stop cassette, resistant gene cassette. Only when you breed this to a specific Cre driver that this downstream gene such as APOE will be expressed. This cassette also has a built-in RS-GFP. So the cell express APOE will have green fluorescence. So this is important for us when we want to do live cell imaging, for example, monitoring microglial dynamics. This is also have a built-in TED-off system, so that when you add doxycycline, you shut down the gene expression. I also want to emphasize this is not a transgenic animal, it's a knock-in allele to the mouse ROSA locus, so there's one copy per genome, whatever gene we want to put in there. So really we bred this to GFAP Cre, which drive the expression primarily in astrocyte. Normally in the brain, more than 90% of APOE is produced by astrocyte. And really want to ask question, well, how does it impact the amyloid pathology of express either E3 or E4? And comparison typically is litter-mate, Cre minus and Cre positive lines. So basically we designed this experiment expressing APOE throughout the 9 months period in these APP-PS1 amyloid model mice, or only during the seeding stage. In these animals, really the first 6 months, amyloid were seed, or during the so-called rapid growth period, not expressing during the seeding, but really expressing E4 or E3 only in the rapid growth period, and really look at the outcome on the amyloid plaque. So you can see that this is just sort of validated what others have published, that if you express in this case, an overexpression of E3. It does not affect the amyloid, but if you do that with APOE4, the amyloid amount significantly increase both in cortex and hippocampus. But this is very interesting here that if you express APOE4 during the seeding stage, you can see that cortex hippocampus, the amyloid pathology pretty much doubled. Okay. But if you express not during the seeding stage, but only in the rapid growth period, you don't see that. Actually, if anything, you see a trending decrease of amyloid. Okay, so this really, allow us to conclude that the E4 really drives amyloid pathology by promoting seeding, and in that period, it's likely, impeding the A-beta clearance, which have shown the clearance is slower, and also promoting the aggregation. So Dave Holtzman's lab had a back-to-back paper with us where they use the ASO knockdown technology, but found the same result, that is only if you knock down APOE4 early that is effective in reducing amyloid. So amyloid is the main pathway, but not the only pathway, not the whole story, because there are a lot of other pathways involved. And then clinically, you can see this because I mentioned earlier, at least in some cohort, that E2 is not necessarily that much protective against amyloid when you compare to E3 in the presence of another allele being E4. However, if you look at the clinical age at onset, E2, E4 does have a delayed age of onset over 2 years. So this suggests that E2 have protective function may be independent from the amyloid. And we can also see that in looking at the large clinical cohort. This is a paper we published a few years back by looking at the National Alzheimer Coordinating Center early data from over 30 ADRCs around the country, and just look at their cognitive decline. And you can see that if you look at the APOE allele effect, E4 had the steepest decline, typically start about age 60. Okay, so some people when they're 70, 80, they say, hey, I still have the same memory. These kind of data says otherwise. But here, more importantly, you can see if an E3 allele, this allele function, you have a slower decline of memory. This is obviously cross-sectional, but if you have an E2, it's actually protected. Okay, and if you separate it out as genome type specific effect, you can see that the E4 effect is dose dependent. This is E3, E4. This is E4, E4. Okay, so E2 protective is also dose dependent. If you look at the subcohort, very smaller number now, without detectable amyloid, you actually also see the same relationship, E4, E3, and E2. Obviously, these curves are not smooth anymore because of the smaller size. And we can completely reproduce this in animal models. So these animal models are what we call the APOE targeted replacement mice, where human E2, E3, E4 allele are knocked into mouse APOE locus, right? Then we perform these so-called memory tests, Morris water maze test. So without going into detail, and this is really training to find the target, and then challenge the animal for memory recall by swimming around this target quadrant. So you can see that if you challenge ask for 24-hour after training recall, you can see that both E2 and E3 have better preserved memory than E4. By the way, these happens only in older animal. If you look at the young animal, we don't see this genome type dependent effect. However, if you challenge the 72 hours after training, even much more challenging for them to know where the target is, only E2 have preserved memory, not E3 anymore. Okay. So we kind of follow up by looking at some of these models and really found a few things. One is APOE level seem to separate out the APOE genome type effect, specifically in cortex hippocampus and also CSF and plasma. E2 always have the higher level than E3 than E4. And this APOE level actually correlate with the memory performance. And this is cross genome type, just have enough N number, both 24-hour and 72-hour memory recall. Also, if you look at the cholesterol level, we're actually a little bit surprised at the beginning that the E2 animal actually has the lowest cholesterol level. And this is looking specifically at the cortex, the gray matter cortex. E2 had the lowest, and then E3, then E4, even though it's not significant. What's interesting is that the cortex level is inversely correlated with memory performance. That is the lower cortical cholesterol correlates with better memory performance. And also the cholesterol also correlate with the APOE level. So what we think is, this is the CSF cholesterol. What we think happened is that APOE has a very important function in lipid efflux, and some of this obviously gets transported to CSF as a place we can detect the lipid distribution, but some of it get transported to whatever synapse or perhaps myelination and so on, where lipid is needed. So we also a paper we published a year and a half ago that we show that the similar thing, APOE and cholesterol level correlate with preserved activity and longevity using the same set of animal models. So more recently, we've done some unbiased omics study, and much of these studies are still ongoing. And what we want to do is really take these APOE target replacement animal, E2, E3, E4, male or female, and then at the young, middle, and old age, and really profiling them, either brain or the periphery serum, transcriptomic, either bulk or single cell, proteomic, metabolomic, lipidomic, and really look at what can we look at unbiasedly pathways that are impacted by APOE genotype, and obviously it's the effect of aging and sex. These experiments are also done with the amyloid background, 5-XFAD. And then in the paper we published 2020, where we reported this result, bulk RNA-seq in the brain and peripheral metabolomic, where we found that actually the aging has the biggest effect on the brain transcriptomic. And you can see that the 3 months animal clustered separately from the 12 months and 24 months. APOE genotype had the second biggest effect. And if we look at what are these modules, you can see that the aging dependent effect, you do see this immune module with your TREM2 and TARO-BP being part of this module. And then this module is actually so-called the extracellular vesicle or vesicular module, where you see that these changes occur in the middle age already, 12 months for all the 3 genotypes. But the immune module only happen in the advanced age, only 24 months, but not in 12 months. However, if you look at the metabolomic of the blood or serum, you can see that APOE genotype had the biggest effect. In this diagram specifically, you see that the E2 cluster on its own, whereas E3 and E4 are very similar. So obviously we have look at the specific metabolites and found some pathways, and those are all described in there. But this type of study really allow us starting getting some picture that, well, in maybe APOE2, some of the APOE2 protective effect might relate to the metabolism or vascular function from the periphery. And then when we performed the bulk RNA, bulk brain proteomic, we actually also saw a very interesting, interesting observation. That is, if you look at the E2 cohort, 3 months E2, 12 months E2, and 24 months E2, you actually don't see much of changes in gene expression. Whereas if you compare these to the E3 and E4, some of these genes go up during aging, right? So 12 months and 24 months. So some of these genes, E3 and E4, goes down. Whereas the E2, you don't see much of that effect. So somehow E2 had this ability to preserve molecular profile. And I think some of this relate to correlate with some of the phenotype and including the memory performance I mentioned earlier. So obviously we're looking at some of the specific genes and pathways and try to construct a picture and build hypotheses to test them. We also try to look at in the individual cell type, what happens during the aging and its effect of APOE genotype. So to do that, we actually isolate single cells And some of this single cell just subject to single cell RNA sequencing. Some of the cells we do isolation, either the population for microglia or astrocyte. We're still trying to work out the neuronal population and subject them to, for example, proteomics or lipidomics. And so here are some of the proteomic with isolated microglia. And you can see that the aging still had the biggest effect on the different modules, but you also see some module impacted by APOE genotype or sex. And if you look at some of these specific modules, you can see that this 3 module, brown, green, dark red, they're affected by age or aging independent of APOE genotype or sex. However, if you look at the salmon module, which is here, it's actually affected by aging, APOE genotype, and sex. You can see that this, in this case, E3, E4 genotype aging has much greater effect than for the E2 animals. So I mentioned our lab also used multiple iPSC models. So these could be either single cell type, neuron astrocyte, microglia, or more recently, we use a lot of these so-called cerebral organoid model. And I think in California, you guys know this really well. Rusty Gage and others, really the pioneers are in these fields. But these cerebral organoid, we call them mini brain. Actually, they can grow to 4 or 5 millimeter in diameter in vitro. They have layers of neuron, and also ventricles. So really have some primitive structure resemble developing brain. So using those organoid, we start looking at some of the effect of APOE. For example, in this APOE knockout isogenic line, we saw that APOE lipid, which detected as lipid droplet by this BODIPI dye, and you can see that accumulate in APOE knockout cells. And then also this marker PLIN2 is showing a significant increase. So this really confirmed the important role of APOE in lipid efflux or lipid metabolism. And actually, if you do this with APOE4 line, you get similar result, not as dramatic as APOE knockout. In that case, APOE4 likely has a loss of function. And one other observation, which we published last year, was that the APOE knockout line also have accumulation of synuclein. And we think that's related to lipid as well, but we're working out some of the detailed mechanism. So really, I think the way APOE have differential effect in probably mostly non-amyloid pathway, it has to do with lipid metabolism. And then we think APOE2 is hyperlipidated, or it's a better lipid receptor, and therefore can distribute lipid in a better way, supporting synaptic and cognitive function. And also when APOE is lipidated or highly lipidated, it actually reduces A-beta aggregation. So some of them might be translated to changes in amyloid. So I think many of you know that lipid metabolism, lipid droplet, receiving a lot of attention in the Alzheimer research field, and I think APOE function is actually quite central in this process. So the other kind of story I want to tell is APOE aggregation. So A-beta aggregate in Alzheimer brain, tau aggregate, but APOE also aggregate. And much of this is from an APOE protective variant. So where do we see APOE aggregation? In addition to amyloid, we also see APOE aggregation actually in endosome. Some of the other investigators, such as Yakin Pers and others, have shown similar results. And that is when APOE E4 in particular aggregate, typically in an age-dependent manner, it co-aggregates some of the important molecules, including receptors. And in this publication, we show that APOE4 actually aggregate insulin receptor, and that's a mechanism to impair insulin signaling. And others have shown that it's impair the function of APOE receptor 2, glutamate receptor, and so on. But this protective variant, used to be called a V236E, which is the amino acid change. We renamed this in the most recent paper of APOE3 Jacksonville variant. It is quite rare, but it is protective in the population or the cohort we have, and then also validated by some of the other studies. But early in this paper we published, we found that the way APOE Jacksonville variant reduce Alzheimer risk is by reducing APOE aggregation. And you can see that if we make APOE, this is just expression in HEK cells, normally APOE, this case E3, come out as a tetramer, and because the APOE self-aggregate. But if you introduce this mutation or variation, P236E, you can see this is mostly monomer. Maybe it has some tetramer, but mostly monomer. You can do cross-linking. You can see that E3 is mostly cross-linked to tetramer, but this variant, Jacksonville variant, is primarily monomer. The reduced aggregation also translates to better lipid acceptor. So in this cholesterol efflux assay, you can see that this variant has better ability to accept cholesterol in this ABCA1 dependent lipid transport assay. It does not affect the receptor binding. We then use the viral mediated expression system to see if it affects amyloid, and the short answer is yes. We do see a reduction of amyloid plaque or the insoluble A-beta 40 or 42. Also, the plaque-associated APOE is decreased, and I think this reflects the reduced aggregation of APOE. And this reduced plaque-associated APOE also correlate with reduced number of microglia, okay, which is shown here, because I think the microglia clustering around the amyloid, some of this probably depend on the presence of APOE. This is also reduced the plaque-associated dystrophic neurite, as detected here, and also correlate with plaque-associated APOE. So the last part of my talk, I want to just talk a couple of stories about the APOE expression in other cell types beyond astrocyte. Because APOE is really expressed by many different cell types, both in the brain and also in the peripheral. So in the brain, as I mentioned, 90-some% by astrocyte, also highly expressed by activated microglia. Microglia normally don't express much, but when they're activated, APOE expression goes up significantly. And maybe to a lesser extent when neurons either get stressed or injured. And in the periphery, which has about 10 times higher concentration than brain APOE, if you compare plasma versus CSF, APOE is produced primarily by liver hepatocyte, but also macrophages. They express large amount of APOE. And then finally, in the vasculature, APOE expressed primarily by so-called neural cells, smooth muscle cells, and parasites, and to a lesser extent, endothelial cells as well. So how do we tease out these individual function of APOE in different cell types? So we really, first want to understand what's the function in microglia. To understand microglia, we went back some of the old literature, and this is actually by Bob Maley. This is really from many years ago. I still enjoy reading his review article. This is from 1998 in Science. Basically, in this review article, he cited the experiment where really the group just performed the acute injury, sciatic nerve, where you can attract the sort of these macrophages, tissue macrophages come in, and really have vastly upregulated APOE expression, hundreds of fold. And these APOE, when they secreted, they would reorganize these lipid components from the injury, and then taking into macrophages and form these lipid droplets. Okay, so lipid droplet concept is obviously not new at all, right? So we're thinking about his macrophage function. And then during the repair process, these lipid droplets will be transferred to these so-called Schwann cells and used for the remyelination process. So this, we think a similar effect or function of APOE also true for microglia, even though we don't have the all the evidence for that. But just in general, to tease out the APOE function in microglia, we took advantage of that conditional cassette that I introduced earlier, except now we bred this animal to CX3CR1 Cree mice, which is tamoxifen inducible animal. So this will drive the expression in microglia. These animals were bred into APOE knockout background so that we don't have APOE expressed except in these microglia. And then these were either treated or not treated with tamoxifen, either in the absence or presence of amyloid background. So when we first just look at the morphology of these microglia, and remember there's a GFP in the cassette. So these microglia, we can just look at their morphology. You can see that the E3 microglia looks somewhat different than the APOE4 microglia. And these microglia are more homeostatic-like, and E4 almost look a bit like a dystrophic. And we can quantify some of these parameters, defining them. For example, if you look at the total branch lengths of these processes of microglia, E3 have longer. Branching point number, E3 had more. So, this further validate that observation at the morphological level. The E3 microglia also seem to be more dynamic. And then some of you might have heard or read the work of Longjun Wu and others that microglia actually has surveillance function, in particular when neurons are not as active. For example, when animals are anesthetized or sleeping, these microglia become very active in terms of sensing what's going on in the environment. So if we use a 2-photon to monitor the dynamic of these processes, and by measuring the velocity of extension or contraction, you can see that the E3 microglia have better or faster velocity compared to E4 microglia in these processes dynamic movement. So then we look at perform the so-called laser-induced injury. So this is actually a little movie. You can see if we perform injury in this area, this is the laser-induced focal injury. These microglia will extend processes and then really respond to this in a very rapid and robust manner. If you do this with APOE4 microglia, you see similar event except to a lesser extent. And you can see that then, see them coming here, but not as much as APOE3 microglia. So can we quantify this? And so, so if you quantify this over a period of 30 minutes, you can see that E3, E4 quantify, E3 have greater response than E4 microglia. We also measure the just behavior, and we detect a slight better performance, at least in this contextual fear conditioning test for E3 microglia animal, not in this Q test. Whereas E4 microglia, we did not see that effect. We also measure electrophysiology, measure specifically LTP. You can see that E3 actually enhance LTP, and E4 actually impair LTP. And some of this, there are some literature about how microglia actually involve in synaptic function, even though not all the molecular pathways are understood, but we're pretty interested in terms of having this observation and then go further to try to understand the mechanism. We then bred these animals to the amyloid background, and you can see that in this E3 expressing microglia, they actually reduce the amyloid both in the cortex and hippocampus. But if we express E4 in microglia, we did not see that effect. So we think this is E4 is a loss of function effect, or perhaps locking microglia in a state where it does not have the beneficial effect, for example, phagocytosing amyloid. And, this is also reflected in the insoluble A beta, E3 microglia reduced, E4 did not have effect. And then also, if we look at the microglia cluster around the amyloid plaque, so this is, you can see that the E3 microglia have a much more robust clustering compared to the E4 microglia. So these are the red are indications of microglia. As you can see, E4 have less and quantified in here. If you measure the dystrophin neurite, you see E3 reduce it, E4 did not have the effect, actually trending to increase. You can actually, these are most likely the one clustering around the amyloid are phagocytic microglia, because you can see that we can capture these microglia in act, so to say, because you can see that some of these microglia have the phagocytosed amyloid plaque, which are actually stained by a dye, quantified in here. We also then perform the single cell RNA sequencing. Really want to start getting some idea about, what are these microglia when they express E3, E4. So in this single cell, using 10x genome platform, we saw 9 clusters of microglia. And then, they're identified by specific marker. You can see some of these are clustered for so-called homeostatic microglia. Some of these are activated microglia. There are also a cluster called the cycling and proliferating microglia. And you can see that if you look at the cell numbers, cluster 7 and 9, E3 actually significantly increased them, whereas E4 did not have much effect. If you look at the differentially expressed gene, also cluster 7 and 9, E3 and E4 both have effect, but they look quite different. If we look at specifically examine the population of so-called activated response microglia, or ARM, we found that E3 significantly upregulate many genes, whereas such defects are much less by the APOE4. Specifically, the so-called immune antigen presentation pathway goes up by E3, the homeostatic genes goes down. So this is really, maybe promoting the microglia clustering, including some of those phagocytic microglia. Whereas E4, you see the increase of, TNF pathway and some of the other inflammation-related pathway, reduced complement and reduced TREM2. If you use the DEG to construct the pathway, you can see that E3 upregulates some of these pathway antigen presentation and so on. E4, whereas it downregulates some of these genes, including complement pathway and some of these other important pathways. So the last just couple minutes, just want to show some data about the periphery effect of APOE. And I think this, as I mentioned earlier, APOE is actually expressed, detected at 10 times higher than CSF. And E4 is actually also a strong risk factor for hypercholesterolemia and atherosclerosis. And so we basically want to ask questions, if APOE4 in the periphery have an effect on what's going on in the brain in terms of cognition or Alzheimer pathology. So to do that, we went back to this conditional mouse model, except now we bred them to the albumin-free as a driver. So this drive expression in liver hepatocyte, either with or without amyloid, comparison between CRE minus and CRE positive. So here, just some validation. You can see that CRE positive, you see liver APOE, plasma APOE. You do not have detectable APOE either by ELISA or Western blot in the brain. So, not saying there's not a few molecular APOE, but at least not detectable level. So E3 or E4, they can both rescue this so-called hypercholesterolemia effect. You can see that the knockout or the CRE-negative at this higher level VLDL, which is part of the phenotype of hypercholesterolemia, but expressing E4 bring this back to the target replacement animal profile. If you look at the effect on the memory and synaptic plasticity, you can see that E3 expressed in the liver actually enhance memory, whereas E4 actually impair memory. Okay, so contextual acute fear conditioning did not have effect on the anxiety or motor function. E3 actually enhance LTP, E4 reduce LTP. So we're really excited that, well, expressing APOE3 or E4, they have differential effect on what's going on in the brain in terms of cognition. So how does the peripheral APOE does that? We think a primary pathway by APOE, peripheral APOE, is the vascular system. And one of the observations we had, some of you might be familiar with the Pathobiology work, where E4 caused blood-brain barrier leakage. We actually saw a similar effect. If E4 induced in the periphery, you can see greater leakage. Here is detected by 43,000 dextrin, measured by 2 photons. We also saw reduced blood flow and then some level of degeneration during the adult, in the adult brain of some of these blood vessels. So what about amyloid? So if you brought to the amyloid background, E3 actually reduce amyloid, E4 enhance amyloid. And that's also shown by insoluble A beta when we grind up the brain. Did not have much effect on some of the synaptic markers, but except in the E3, you seem to enhance the presynaptic marker a bit. But it does also change the GFAP, which respond to amyloid. So we also use a complementary technique, and that is to use young plasma from either E3 or E4 animal to inject them to older wild-type animal. And some of you might be familiar with Tony Weiskor's work, where he showed the young plasma actually has a rejuvenating effect. So we basically want to ask question whether if you use E3 or E4 young plasma. But basically we saw E3 young plasma has the effect to improve the cognition, whereas the E4 did not seem to have that effect. Okay. And showing both here and here. Also, if you look at the blood-brain barrier leakage, E4 seem to enhance this leakage, whereas E3 seem to have somewhat protective effect. So these kind of experiments are really interesting, and because of these results, we actually started a clinical trial, and this is a collaboration with our behavioral neurology, Dr. Neil Graf-Redford, and really treating the E4 patient who are in the early stage of Alzheimer, MCI, with the E3 young plasma. And really just want to see, well, one, is it safe? Obviously, it's phase 1. And 2 is, if we measure cognition, do imaging, and measure fluid biomarker, do we see any effect? And also allow us to look at some of the, the dynamic of APOE distribution when we try to switch the genome type, at least in the periphery, to guide a future study. But really, we think the periphery APOE genome type has significant effect on what's going on in the brain. So in summary, lesson learned, astrocytic expression of E4, but not E3, enhanced amyloid pathology by inhibiting A-beta clearance and promoting amyloid seeding. We think this is mostly related to E4 gain of toxic function. E3 protect AD risk and aging-related cognitive decline likely by enhancing APOE function lipid transport. And we think E4 likely have a loss of function. And the E2 and E3 protective variants likely enhance lipidation, partially by reducing aggregation. Microglial specific expression of E3, but not E4, enhance memory and synaptic plasticity and reduce amyloid pathology. And then finally, the liver specific expression of E3, enhance brain cognition, reduce amyloid, whereas E4 have the opposite effect. So how does it guide us in terms of how to target APOE? And obviously this conversation been going on in the field. Do we increase APOE or decrease APOE? But we think it probably should be dependent on the APOE genome type. For example, if an individual carry a E4 genome type, I think the dominant toxic effect of E4 should be controlled, and therefore downregulating APOE, for example, by antisense oligonucleotide or E4 specific antibody might be beneficial, especially in the diseased patient. Whereas the individual do not have an E4 allele, 222333, maybe increase the APOE level is helpful. In particular, the lipid transporting function or APOE lipidation. So we think this is obviously still need some preclinical testing, but really APOE genome type is very important. decreasing APOE aggregation, in particular E4, we think can be helpful. E2 and protective variants, learning from Christchurch, which I did not get a chance to talk about, or Jacksonville variant can be explored by AAV mediated delivery or through recombinant lipidated protein. And some of the APOE-related molecule, APOE receptor, either beneficial or detrimental could have effect. Can we do individualized treatment for APOE? The answer is yes. If we look at APOE genotype distribution in Alzheimer patients, these are different cohorts. You can see that the E44 homozygous account for about 15% of the Alzheimer population, 10 to 15%. And you have about 40% individual who are E3, E4 genotype. And then you also have about 40% or so, either E2, E3, E3, E3. So therefore, targeting strategy, as what I just mentioned, I think can be explored toward individualized therapy. And just want to acknowledge people in the lab. These individuals highlighted, 3 of them already, assistant professor, either independent or still in my lab, really performed the bulk of these work or led the work I discussed. And these other people contributed, some of my collaborators, previous lab members, and my funding agency. Thank you for your attention.